Hello and welcome to today's webinar. My name is Olga Bodie and I'm part of the team here at Syngap Research Fund. Our presentation today is Illuminating the Dynamic Architecture and Regulatory Mechanisms of Syngap. And I have the pleasure to introduce today's speaker, Eric Underbaki. Dr. Underbaki is an associate professor in the Roy J. Carver Department of Biochemistry, Biophysics, and Molecular Biology at the Iowa State University. His research team investigates the dynamic structures of signaling protein teams that tune neuronal connections, to profile protein communications in action. The Underbaki lab employs integrative structural biology approaches, including mass spectrometry, chemical probes, and enzymology. Eric grew up in Iowa and earned a BS at Iowa State University. He pursued a PhD in biochemistry at the University of Wisconsin in Madison. He developed an interest in the structures of synaptic proteins as postdoctoral research fellow at the University of California, Berkeley, and at the Scripps Institute. He returned to Iowa State University to start an independent research group in 2015. A recorded version of this webinar will be available on the SRF website under webinars on the family menu. And then by the end of this presentation, you will have the opportunity to get the answers to your questions, and we'd love to hear from you. So please write your questions in the Q&A below. And for those of you just joining us, welcome. Again, our speaker is Dr. Eric Underbaki, and his presentation today is Illuminating the Dynamic Architecture and Regulatory Mechanisms of Syngap. Welcome, Dr. Underbaki.
Thank you so much for that introduction. It's a real pleasure to join you all here today. I am going to pop up a slideshow so that I can guide you all through what my research team is exploring in terms of the dynamics and structures of Syngap and some of their signaling mechanisms. So again, thank you so much for this invitation. I rarely get the opportunity to speak to a community of people so invested and active in pursuing new research to treat a really important issue. So I guess I will start off with a little bit of introduction. There we go. I want to introduce my team, the people who do this research alongside with me. And we're at Iowa State University in a biochemistry program. We are largely protein biochemists, and our work is focused largely on the kind of detailed structures of the proteins and their interactions and how they work. And this is a sampling of the people who started the project, an undergrad in my lab named Sarah, and a graduate student who has since graduated and is at the National Institutes of Health now, Quinn Hansen. And these are some current people in my lab, graduate students Tanya and Alexandria, and a small team of undergrads who work with them as well. And collectively, they are exploring a number of different questions involved in mechanisms of synaptic signaling. So, as Olga mentioned in that lovely introduction, I have had a long-standing interest in how signaling systems work, the processes by which cells sense their environment and respond and communicate between each other. And this is a major and complex topic because a lot of the proteins involved in these communication pathways are membrane associated. If not going through the membrane, then they're often clustered next to the membrane. And these proteins tend to have a lot of regulatory features. They're often composed of multiple domains. They often are very flexible and highly dynamic, and they are often at a tipping point of responsivity. They're not really just slammed on or off. They're kind of capable of tuning their activities over a wide dynamic range. And so this makes them challenging targets for classical structure function studies for biochemists. And there are many, many important disease states associated with the dysfunction of these systems. So they're a real frontier topic in lots of scales of biochemistry, cell signaling genetics, and developmental biology. So I've studied several different signaling systems in my career. Most recently, before starting my lab, I, as a postdoctoral researcher, was studying the mechanisms of nitric oxide signaling. And this is a really strange and unique signaling pathway that involves an enzyme making a gas molecule that can kind of diffuse between cells and communicate really locally. And one of the locations in which this nitric oxide gas signaling is important is at the synapse. And so this was my introduction to synaptic signaling and the signaling proteins of the postsynaptic density. So when I started my independent lab, I moved kind of laterally, looking at the nitric oxide signaling proteins and their scaffolds, and looking at the other signaling proteins that were brought into this kind of signaling cluster with these scaffolds. So I probably don't need to reinforce this too much with this audience, but the proteins involved in the synaptic signaling are primarily responsible for two kind of levels of signaling of cell communication. One is the kind of primary release of neurotransmitters and then generation of an action potential. But there's a bunch of signaling proteins in the postsynaptic density, especially, that are involved in tuning that primary response, adapting to patterns of signal over time. And they are responsible for kind of making decisions, if you will, at a molecular level for whether or not this is an important and well-trafficked neuronal connection, in which case it can be strengthened, or whether it's prone to noise and background, in which case it can be kind of tuned down. And so this is a really complex system involving a number of different proteins. They're often connected right next to the membrane through interactions with scaffolds. And one of the most prominent is, of course, Syngap. So there is a wealth of studies and research going on, looking at all sorts of different signaling proteins in these systems, including kinases, G protein signaling, the neurotransmitter receptors themselves, and of course the scaffolds. And collectively, these are really important targets of study because it's just well understood in an era of personalized genomics that mutations that impact the functions of these are associated with a really wide range of neurological conditions, some of which can be quite severe, and some kind of on a spectrum lead to neurodivergences. And so there is all sorts of study happening at all kinds of levels here to investigate how these proteins communicate, how they sense the trafficking patterns, the signaling and action potential patterns of the synapse. And we are building on a multiple decades long quest to understand these systems. And the researchers that are exploring these systems are coming at it from multiple, I guess, scales of complexity. From my end of the world, the protein biochemists, and we are very focused on a kind of dissection approach where we take individual proteins one at a time and we explore the fine details of their structures and functions. And that's been very important and useful for establishing a baseline of what these proteins do in isolation. At the very other end, we have tremendous work that's been done through the decades studying at a more organismal level or at the cell-based level, identifying the proteins and pathways involved in long-term potentiation, long-term depression, and all the other factors going into neuronal communication and synaptic plasticity. And so these are pretty well established approaches to studying the mechanisms of neuronal communication. And both are converging on a frontier that's not exactly new, but is still in need of a lot of work. And that's kind of the mesoscale organization of the proteins involved in this communication. Rather than focusing on individual proteins one by one, there is a increasing attention to how these proteins form scaffolded networks, higher order complexes, and how these move and rearrange and respond to stimulus. And so this has been a classical challenge of structural biology, but happily we have a lot more tools available to us nowadays so that we can explore these bigger scale signaling complexes. I want to take a step back though for a broad audience and kind of, I guess, review some perspective on why we care about the structures of proteins involved in neuronal communication. This field in general is known as structural biology, and it's kind of a descriptive field. We're interested in understanding how the structures of really large macromolecules involved in biology how those structures underlie their functions. And that's kind of an adage in my field: structure leads to function. And a real big goal of this is to take the kind of conceptual cartoons where we describe how enzymes or RNA or any of the other macromolecules of life, how they kind of interact at a cartoon level. And we want to go deeper and understand what those molecules look like at a molecular level. And this has been really important and useful for understanding how they work and how we can address their functions and manipulate their functions, either through pharmacological interventions or other means. So I guess one important lesson that I like to share with students in my introductory classes is that the molecules of life are just molecules. They're behaving the same with the same chemical principles as any other chemical outside that you might encounter in a high school chemistry lab. One major difference, though, is that these macromolecules, as the name would suggest, macro, they're huge, and they have architecture. They have really big cavernous structures with pockets and nodes and projectiles, and this shape complexity allows them to recognize each other, recognize small molecules, and transform small molecules with real exquisite precision. So these real big structures can interlock with incredible specificity. They can change each other's shapes. They can sense and respond to each other, and they can modify each other. And it's through those recognitions and modifications that we can get signaling and communication at a molecular level. It's also of interest to people trying to modulate that communication, because the giant shapes of these proteins or other macromolecules allows us to develop drugs that can kind of sneak in and fit in those pockets and tune their functions. So this is a long-standing frontier goal of biochemists and biologists in general, understanding the structures of these large molecules. And I like to think about what that really entails, because we kind of take it for granted that this is just something we can readily do nowadays because we've been doing it for 50 or 60 years. But what we're asking of researchers is to describe these really, really huge molecules at an atomic level, looking at the kind of physical location of all of the atoms and how they're connected with bonds. And I remember when I was in college in an organic chemistry class, my professor walked us through the really inspiring work that Emil Fischer did to reveal the structure of glucose, which is right here. It's an ornate and important molecule. It's the foundation of our metabolism. And it took a long time to understand how its atoms were connected and how they kind of projected in three-dimensional space. But nowadays, to understand cell biology, we're actually looking at structures of proteins. And this is just an average kind of small protein, the glucose binding protein. And you can see just how dizzyingly complex this is. It's just a warren of a thicket of different bonds connecting the atoms of amino acids. And they kind of come together with this global architecture that's able to do things like, in this case, bind to and recognize glucose. But that's not all. There's been a lot of focus on the structures and functions of the proteins that we can readily capture and use our tools to describe the three-dimensional structure. But there's a whole nother class of proteins that have intrinsically disordered regions. And these are proteins that don't actually adopt a static, stable structure. And I should say, in fairness, all proteins have dynamics to a degree. They kind of breathe, they flex, they move, but there are huge numbers of proteins that either are entirely disordered or have significant regions of disorder where the structures of these are whipping around and capable of moving a lot. And so a colleague of mine, Julian Roche, calls this the kind of dark matter of the population of proteins in cells. So we have overlooked them for a long time because they're very hard to study. And so there's kind of a natural bias to focusing on the structures of the proteins that have structures that are easy to study. But indeed, most proteins, especially signaling proteins, have a lot of disorder in them. And it's important to recognize that and describe structures and signaling pathways in terms of that order and disorder. So this brings us to some of the approaches my lab uses. We study a number of different proteins involved in synaptic communication, and they all share certain challenges that make them resistant to classical structural biology approaches. The details aren't super critical, but we have things like X-ray crystallography and nuclear magnetic resonance and cryo-electron microscopy that help us understand at a really, really high resolution the shapes and functions of these biological molecules. But not all proteins are easily explored using those classical techniques. And signaling proteins are really good examples of that problem. They're often composed of multiple different subdomains, regions where the amino acids fold into unique shapes with unique functions, and they're all strung together, often by very flexible linkers that can be very long and are usually to some degree intrinsically disordered. And so these proteins move around. Essentially, they don't sit still for the photo, and they have resisted a lot of the structural characterization that we take for granted in other systems like metabolism. They're also famously transient in their interactions, meaning they kind of lightly contact one another and they fall apart, dissociating from one another very easily. And that's a feature, not a bug. Signaling and communication is all about kind of light interactions, assembly, communication, and then quick disassembly when the signal is no longer needed. So it's the reversibility of this communication that's really important, so we don't lock the cell into a particular mode. We want to be able to reverse this all and easily. But that is, again, a challenge for getting these things to kind of assemble, hold still, so that we can structurally characterize them and how they work. So we do have a lot to rely on, though. We have the wealth of work of geneticists and cell biologists who have identified the interconnectivity of these different proteins, the importance of what they do in a cell. And at the other end, we have the structural biologists who have, through the decades, broken these difficult proteins into little pieces and structurally characterized the pieces. And so one of my lab's motivations in studying how signaling proteins work at the protein level is to acknowledge that we do have a lot of great high resolution structural information describing important proteins, or at least pieces of these proteins. And our big question is putting those pieces together and describing a higher order architecture of how these assemble together and communicate and move, with a strong focus on understanding that there isn't a single snapshot to be had here. These are rapidly moving proteins that kind of quickly assemble and disassemble, and they sort of flex around each other, and they interlock in a big meshwork of other proteins. So we need tools that aren't necessarily going to give us a single unitary, locked in structure, but rather tools that can describe an ensemble of structures, the range of motions available to these proteins, trying to capture them in action. And so a kind of newish and exciting approach to this is called integrative structural biology. And that's where we kind of try anything we can to get restraints on the structure, little details describing the range of structures available and possible. And then we use computers to try to put that together to build a model of how a really complex array of proteins can kind of come together and do their job in a cell. And my lab focuses on a collection described in cartoons here. And I won't drag you through the details of how all of these biophysical things work, but the higher order, or the big picture idea here, is that we do have some reliable models of individual pieces of our signaling complexes, either through a history of structural biology or through really neat machine learning algorithms that can predict with uncanny accuracy what the structures will be. And then we ask how those pieces fit together and move around each other using different techniques. Some of them are based on X-ray scattering. Some of them are based on just basic biochemistry, where we try to generate mutants and see what happens to the functions of these individual proteins. But we also take advantage of a lot of mass spectrometry based tools. And this is one of the things that makes my lab a little bit odd. We do a lot of mass spectrometry to study proteins. And that is a technical sounding word, and we don't necessarily need to dissect what a mass spectrometer is. But I want to acknowledge that mass spectrometry is in its own right a really, really useful tool for a lot of approaches to studying biology. And I want to give credit to the people who developed this technology over the years. In general, a mass spectrometer does a pretty straightforward job. It's able to ionize a molecule and then measure the mass to charge ratio. And what that comes down to is that we get a really, really high precision mass measurement of individual molecules. And with that mass measurement, we can identify what molecules are in a cell with great precision, and we can survey whole populations of proteins within cells and ask how those protein populations change under different conditions, either with a mutation or maybe in a cancer cell line or in response to different developmental stages. And so mass spectrometry has become an absolutely foundational technology for proteomics, which is the field that is responsible for telling us what proteins are doing what in what cells. So that's really important. And I know there are some excellent proteomicists working in the Syngap field right now. So that, I think, is what mass spectrometry is most famous for, is profiling what proteins are doing things in cells. And so it's a little unusual for mass spectrometers to be used to study not the mass of a protein, but rather the structure of a protein. But we've come to embrace mass spectrometry to do this because it brings in certain advantages, as long as you apply some tricks to going beyond just the mass per charge ratio. And so the details are kind of technical, but there are two versions of this that my lab employs. One of them is a isotope labeling of proteins that allows us to paint the surface and try to identify what's exposed and what's dynamic and contrast that with what's buried. And it's called a hydrogen deuterium exchange mass spectrometry, and I'll show some examples of that later. There's also cross-linking approaches where we can try to ask which regions of proteins are in communication with each other, or which ones are close to one another, by kind of capturing them with a cross-link, a chemical bond between these two pieces, so we can then chop apart and identify by mass spectrometry. And between these two approaches, we can learn a lot about the dynamics and higher order architecture of different proteins. So those are the kind of approaches that my lab uses, and we're currently looking at kinases and scaffolds and other things in the postsynaptic density and exploring their structures with mass spectrometry and other integrative structural biology approaches. But very recently, we were fortunate to be awarded a grant to start a pilot study looking at Syngap. And so my big I guess cautionary note here with the rest of this conversation is that we're at the very, very early stages of this research. We have just begun to explore these aspects of Syngap biology. But I was really excited to tell you about where we're going with it and to get your input and hear your questions as well. So a bit of background that many of you probably know better than I do. Syngap has a really complex role in tuning synaptic plasticity and development in neuronal systems. It is named for a central domain in it, the GTPase activating domain that gives it gap activity. But as contemporary research has shown, it has many, many more roles than that. It is not simply a protein that regulates G protein signaling. And we'll kind of march through some of the aspects of those functions in a bit here. But I guess I should reinforce, just so we're on the same page, what it means to be a GTPase activating protein. And that refers to an activity that tunes a family of signaling proteins that are really small, and they tend to skate around the membrane. And they are often initiators of long, elaborate signaling pathways that can often communicate all the way back to the nucleus and control gene regulation. And so RAS and RAP are two GTPases that communicate, and they do so based on whether or not they're off and bound to a small molecule GDP, or on when they're loaded up with the activating GTP. And so there are proteins, GTPase activating proteins, that cause them to hydrolyze their GTP and turn themselves off. And there are guanine nucleotide exchange factors that swap out the inactive GTP for a GTP and turn them back on again. And so initially, Syngap was identified as a gap protein, down-regulating the communication of these GTPases. But as was immediately noticed in the first description of the sequence of that big Syngap protein, it clearly had the capacity to do much more. It had multiple different domains and a really long, apparently intrinsically disordered region that could do many more things in the postsynaptic density. And so that kind of structural and functional complexity is what drew my lab's attention to Syngap, in addition to the obvious, very serious clinical importance of its function and dysfunction. And as we were kind of monitoring and following along as researchers were exploring Syngap function, we were really astounded by a couple facets of Syngap biochemistry and biology. And so here is just kind of a list of some of the things that caught our attention that made Syngap a really, really strange and fascinating protein. And it's really something that we kind of wanted to explore as well. The first thing that caught our attention is that Syngap is, of course, one of the most abundant proteins in the postsynaptic density. And that's very strange and unusual for a protein that's annotated, in, at least in terms of its gap function, as being a regulator of another protein. Hypothetically, these are enzymes that can shut off or tune down the activity of GTPases like RAS and RAP, but you don't really need the gap to be in huge excess of the GTPases to do that job. You just need a couple of them. You could consider the gap proteins as kind of management, kind of dictating what's happening with a bunch of other squads of employee signaling molecules. And you rarely see an excess of the gap above its substrate GTPases. And so, of course, that quickly led to people examining its scaffolding roles, like maybe it's serving a structural role in the postsynaptic density, like PSD95, another well-characterized scaffold in there. And of course, lately, there's been some really astonishing and fascinating developments looking at how the very abundant Syngap and its interactions with PSD95 scaffolds can cause kind of hydrogel formation inside the postsynaptic density, or liquid-liquid phase transitions. Another really curious thing is that there's been a lot of study about what the target of the GTPase activating activity is in Syngap. And so there's been some back and forth about how good it is at tuning RAS signaling versus RAP signaling and what factors are involved in the specificity of the signaling pathway. And so there's been some great work from Kennedy and coworkers and others looking at how kinases can kind of modulate the activity and the preferences for the gap activity. But there's also some really cool new work, and I hope you had a chance to see this. I think on Monday, Huganier and coworkers had a really cool study where they were able to surgically kind of ablate the gap activity of Syngap and showed that there were some really fascinating effects. The primary roles of Syngap apparently are not just as a gap protein, but the dominant role seems to be that scaffolding and that liquid-liquid phase transition that it's doing to regulate access to binding sites and to control how many neurotransmitter receptors are clustered at the postsynaptic density. So this is a really cool paper that just came out as a preprint and really adds some complexity to the picture of what Syngap is doing. From a structural biology point of view, I think you're probably all used to seeing a map of the pieces and parts of Syngap. It's a really big protein, and it's got that central GTPase activating domain that it's named after, but it also has some accessory domains that show up in lots of signaling proteins, and it has a lot of these intrinsically disordered linkers. And so this is just a picture from AlphaFold, that machine learning algorithm for predicting structures. And I don't mean to propose this as a legitimate structure here. It's just, this is what sees intrinsic disordered regions. It just kind of makes this spaghetti wad, but it really gives us a perspective on how much of the total sequence of Syngap is a really dynamic, wavy, intrinsically disordered region. And this disordered area is very, very intimately involved in forming those scaffolding interactions and phase transitions in the postsynaptic density. So my lab is also interested in like these auxiliary domains and what they're doing. There's a domain called a Plexton homology domain, which was identified mostly by sequence conservation. It has some of the kind of sequence features you would expect from a pH domain, but it doesn't apparently fold reliably into a pH domain. And there's still a lot of questions about what it does. Maybe ancestrally, it came from a sequence that folded into a pH domain, but maybe it diverged over evolutionary time. It also has a C2 domain, and C2 domains are famous for binding and responding to calcium, which is intriguing because there's a lot of synaptic signaling involved in calcium flux. And there was kind of interest initially in considering that Syngap might be calcium responsive, but while structural predictions suggest that the C2 domain does have a pretty classic C2 domain fold, it doesn't have the actual residues that would be responsible for binding calcium. So it seems to not respond to calcium. And there's some really cool structural work that suggests that the C2 domain is involved in selecting or nestling into the GTPases that Syngap might tune activity of. So yeah, this is just a whole list, a laundry list of things that remain unclear about the structure and function of Syngap. I think it's been pointed out before that despite many of these regions not having obvious structure or function, they're still quite conserved. They're surprisingly conserved in mammals, and they're even conserved across birds and reptiles and things like that. So there appears to be some long-term selective pressure for maintaining these unusual Syngap features. And this again urges us to explore how they work at a higher level of detail. So my lab and many other labs are interested in these big overall mechanistic questions. For one, what is the dynamic architecture of this whole Syngap molecule? Lately, there's been astounding work showing that it can trimerize and form these condensates, and we'd like to explore how the rest of the domains of Syngap behave in the context of those condensates. There's also longstanding interest in how the auxiliary domains, as well as post-translational modification, influence its enzymatic activity and its scaffolding activity. And I also think it's important to think about Syngap as being a protein that is right next to the membrane, because the GTPases that it does, whose activity it does tune, these are peripheral membrane proteins kind of skating along the edge of the synapse membrane. And so we need to consider how the membrane might be influencing its activity. And of course, the exploration of the higher order scaffolding interactions is very key. Thinking about the competition between those interactions as well. It's probably not just a static web of interactions between Syngap trimers and PSD95, but rather exchanges of these binding sites that are allowing changes in the number of receptors that are in the membrane. And wonderful work is being done in that field as well. And of course, my lab is interested in a lot of post-translational modifications, the kind of molecular flags that change and shift how signaling proteins communicate with one another. And Syngap is indeed subject to phosphorylation, one example of that. Phosphorylation can tune its activity, which usually comes about by tuning its structure or allowing it to interact with new things. So we'd like to explore that some too. So what we had proposed to do was look at a lot of these protein dynamics and structural questions using our kind of oddball mass spectrometry approaches. And these are the two I mentioned before. And it's, I guess, useful to ask, why? Why are we employing mass spectrometry to do this? And the reason is because the mass spectrometry technology itself is really, really easy to apply to humongous proteins and to protein complexes. And that can be a challenge for a lot of the other more traditional structural biology approaches. You can also make these structural measurements in the context of membranes, which are themselves very fluid and challenging for structural biology. We can build up from the bottom up, from purifying multiple different proteins and putting them together and see how they assemble like Legos. And we can kind of probe those bigger structures as well using the same techniques. And we also don't need these proteins to sit still. They can continue to flex and twirl and move around each other. And these mass spectrometry techniques are able to capture the changes in dynamics associated with the linkers and intrinsically disordered regions of signaling proteins. So we're just at the very beginning of this. We just got the research grant 2 months ago, but we are excited to apply these approaches to Syngap, and we have a lot of different questions that we'd like to look at. In our remaining time, I kind of want to give you just a slight glimpse of what that would look like. We won't go into this too much here, but I wanted to show you an example of hydrogen deuterium exchange mass spec. This is considered a footprinting approach where we can use a mass spectrometry readout to examine the dynamic surfaces of signaling proteins and look at how the surfaces interlock and how they move and how they change when they're modified with phosphorylation, for example. And so it involves taking a feature that's universal in proteins. Proteins are made up of individual amino acids, like this one and this one, that are linked together in peptide bonds. And every one of those peptide bonds can chemically exchange with the water solvent. And so this is happening constantly. All of your proteins and all of your cells are exchanging their atoms, specifically the hydrogen atoms, with the water in the cell. But we never really notice it because it's just a hydrogen exchange for hydrogen. But we can actually measure that rate of exchange, how quickly that happens, by using isotopically labeled water. And this is just a heavy isotope that's not radioactive. It's just got one extra neutron. But that one extra neutron is something you can easily measure with mass spectrometry. And so we can profile where in the protein it readily exchanges with the water surrounding it. And that can tell us what's on the surface and what's buried in interactions and what's moving really easily. So here's kind of a cartoon version of that. This is kind of an example protein. And if you dip this protein into isotope labeled water, you can see exchange all around the surfaces, wherever it's kind of moving and dynamic. And we can profile proteins and learn a lot about their structures using the rates of those exchanges. And some of the things we can learn about are kind of substructures that we call secondary structure. We can learn about how quickly different parts of the protein are moving. We can map how proteins interlock and recognize each other, which is important for communication. And we can learn things about the subtle local chemical environment, which would be perturbed by a phosphorylation, for example. And at the end of the day, if you skip past all of the mass spectrum detail, what we get is data that we can color code. And this is popular in my lab. My students like to look at these things in color code terms. It's much easier than just looking at tables of numbers. So this is kind of just a generic example of some data that we took. These are two different kinases that we were exploring. And these are the structures of them. And we kind of knew the static structure from old structural biology approaches, but we wanted to ask what parts were dynamic, like we can get a more action shot of these things. And so we did some hydrogen deuterium exchange mass spectrometry, and we could kind of map over time where exchange happened. And we color coded that going from low exchange in blue to high exchange or high dynamics in red. And you can kind of see the periphery and the edges and the loops exchange and get redder over time. And that's the kind of data that we can extract and use to build models of signaling function. So this is just a cartoon to recognize that these are comparative experiments. We can look at a protein in one state and look at it in another state and compare the dynamics. And so we might look at a protein that's not bound to a partner, like a scaffold, and one that is bound, and we can look at how the dynamics change. And so currently we're we've already started that looking at Syngap and how its exchange rates tell us about interactions. And we began by looking at those auxiliary domains. If we compare a Syngap with its C2 domain cutoff or the whole Syngap, we can examine how it interacts with the G protein. And indeed, we can show the regions color-coded in blue here that are impacted by the binding and association of one of those G protein substrates. And interestingly, even though we think of the gap domain as being the heart of that activity, we see most of the changes in the C2 domain, which was predicted by the structural biology group that initially co-crystallized these together. So we're also looking at how membrane interactions would tune the recognition of Syngap with its substrates. And that's important because RAS and RAP are both GTPases that anchor to the membrane. And so the membrane often has a really important role in that. So I think, just give you some quick examples of what we're moving towards. I won't go into the details of this kinase activity for time's sake, but we were doing very parallel experiments looking at a kinase involved in synaptic plasticity. And we were able to look at how its different domains contacted each other in the same way. And again, we love color coding. So we were able to map how a regulatory domain interacted with the signaling domain. And we could see an imprint left by their interactions where the exchange rates were slowed, and we color code those in blue, and we were able to build a model of that interaction. And then look at how things like phosphorylation change that interaction. And so we see these blue kind of stabilized regions of the higher order protein totally reverse themselves when you phosphorylate, and now they swing into extra dynamics and more readily exchanged. And we could go zooming in real detailed and learn about catalytic mechanisms and signaling mechanisms through that. And so I wanted to make sure I had a time to meet some of you and hear some thoughts and questions. So with that, I kind of want to, again, acknowledge my team. Quinn and Sarah are the two that began this project, building up our Syngap infrastructure, and the ones who interactions with GTPases and profiling the dynamics of that tail region. And want to acknowledge the future colleagues working on this, 2 grad students and a new team of undergrads, and we're recruiting a new person just at the end of this month, and the funding source that we have to support that research. Most recently, the Syngap research comes from the NIH and IMH. So I would welcome any questions or thoughts.
Thank you so much. Yep, let me grab up. I don't see any just yet, but if anybody wants to ask any questions, you can type them in the Q&A or on Facebook in the comment section. And then Janie Reed is here too. She, I know, has some questions, I'm sure. So go ahead.
Hi, Janie.
Hi, Eric. Thank you so much. This is so incredible. And Lauren, you can see the questions, right?
Yeah. Do you see any? No, I just don't see any yet. But yeah, I can.
But you see
I'll
where
let
to
you
look
know.
even? Is it going to be in the chat
Yeah.
and Q&A and open chat?
Yeah.
Okay, I'll
Yeah,
open
I
it
just
both.
don't see any. I got you.
So-
Yeah, I'll let you know.
Oh, Mike's
Okay.
here. Does Mike want to say something before he goes? He's muted.
I am going to run out. I have to run children around, unfortunately. But I was going to say we have a pretty intimate group, although I'm sure this will be watched on video many times. We have some esteemed researchers in the attendee section. So if any of our other Syngap scientists want to ask questions, JR and Lauren, you might just want to promote them to be able to speak. And I just want to express my thanks, Dr. Underbaki, and regrets that I won't be here for the rest of this. All right. Have a good day, guys.
Thank you.
Yeah, so this is JR. This is Lauren, please promote any of the scientists that I think maybe Dr. Wilsey's here. I don't know. I can't actually see who's in. So
Yeah, I got her.
there. So I think my first thing I want to do is just give you so much thanks. This is incredible, like absolutely incredible, like exactly the kind of structural information we're going to want and need. I think one thing that's really interesting about Syngap Syngap 1 related intellectual disability is that it's really considered a haploinsufficiency, and so I can't wait to see all the understanding of what Syngap's doing, but I also kind of am interested in how half of, just having half of it around makes a problem. And then in addition to that, for the missense, how does having one amino acid change out of this whole thing make it look like, it seems like maybe looking like a haploinsufficiency. So or maybe it's not maybe those aren't haploinsufficiencies. And will there be a way that you can tell between a null, a isomorph, a hypermorph, neomorph, and a dominant negative, which are kind of maybe there's more categories than that, but those are kind of like the 5 big ones that I think about.
Yeah, that's a excellent observation and a really profound question I think is being addressed community-wide. Our role in that is only a fraction of it. I do think the last 10 years of work that have shown that, obviously a haploinsufficiency dominates this, and that isn't the kind of thing we would expect from maybe an enzyme that was just controlling GTPase signaling. It's clearly something broader than that, because there is such a sensitivity to the population of stoichiometry within the context of the postsynaptic density. And I think the more recent work showing scaffolding roles, phase transitions, and that neat slot idea, controlling how many neurotransmitter receptors are available to cluster at the PSD as opposed to diffusing through the membrane. I think those are really revelatory in terms of why you see this range of consequence when there is haploinsufficiency. There's also the really interesting notion of point mutations, single residue mutations that can cause very big effects, which is, on the contrary, not what you might expect if this was doing just a scaffolding role. And so it seems to be an interplay of a couple different things, and maybe they work at different timescales and at different stages of development. Certainly, my lab alone is not sufficient to answer that. It'll definitely be a community-wide effort to explore those things. But in addition to what the rest of the community is doing, labs like mine that can look at a structural level are very, very interested in this, looking at how individual point mutations can change the global dynamics or local dynamics or the functions of the gap domain. There's a really cool detail in that new preprint too, about personalized genomics, identifying actual mutations in gap domains that don't appear to have a function, even though you would think that they would have a severe impact on the catalytic function. And so it's research like that that's able to look at the role of the scaffolding, the role of the phase transitions, and compare that to whatever their roles there are for controlling small GTPase signaling too. And so what we're looking at now in my lab are we're very excited to look at missense mutations that are known to have actual impacts on people. And we're also interested in exploring the scaffolding interactions, which is something that we're eager to do. It's especially with cross-linking. There are known differences in how scaffolding interactions change, especially when phosphorylated by CAMK2. And we can see that sort of interaction network that causes the phase transitions can be tuned based on post-translational modification. And that's really important. It looks very much like all the literature is pointing to, that this is not just some sort of static, stable structural force. It's a structuring or a scaffolding that is responsive to patterns of neurotransmission. Sorry, that was kind of a meandering answer, but I agree that those are...
it was I realize it was an unfair question, but that's always kind of forefront in my mind. And I guess I did talk to you before, so I do know that you're going to look at some sense changes, and that's very, very exciting. And there's so many aspects of where your work go that it'll be interesting to see what ends up jumping out first as what you want to look at more. I want to have Dr. Helen Mulsey talk, and so she has a question in the Q&A, and I'll just read it out loud, but maybe you and she can talk. So she says, might have missed it. How are you thinking about cell type specificity of isoforms? Your mass spec approach seems perfect to understand isoforms too.
Oh, that's a really good question. I think there's two levels of that. One would be, I'll give credit to the proteomicists who I know are actively working on this. There are definitely people who are looking at different cell types at different stages of development and using the power of mass spectrometry to identify the abundance. And I'm not 100% sure, but I hope, I expect they're probably also looking at subcellular localization as well, like the sort of patterns of movements of the Syngap. And absolutely, the role of isoforms and different expression patterns of those isoforms is really, really critical to this. And I totally agree that cell type differences are probably very profound, as well as developmental stages. On the other side, my lab is primarily focused on the fine-grained protein details. So we are absolutely interested in isoform differences. We can readily clone different isoforms, and we can sort of produce the individual protein. But that's our take, is usually building up from the sort of molecular level, ground up versus top down. And that's the kind of perspective that we're supplying, but definitely looking forward to both exploring isoform differences, as well as the impacts of known mutations on the dynamics and interactions and scaffolding.
Okay, well, Dr. Wilsey, do you have any follow-up questions on that?
No, that was great. I think it's really exciting. You know, when I think about isoforms and I look at sort of the recent work on all the C-terminal ones, I think they're really interesting, right? And I think sort of understanding how that affects the structure of the protein and then also its interactors, I think could be super cool. So it's great that you're going to take a look.
Absolutely, and I appreciate those thoughts and questions. I share that interest, and it's going to be really, really fascinating to see as different groups focusing at those different scales of complexity come together and reveal how those changes impact that global PSD organization. I think that's just the remarkable thing, especially with some of these phase separation studies, is Syngap is a very central player in that density organization.
Okay, can I ask a pretty basic question?
Absolutely.
About Syngap structure? So there's that really long coiled, that really long coil, and I've heard that referred to as maybe the coiled coil domain. It's near the C-terminal, and I've heard that can form a trimer. How much is actually known about whether it does form a trimer in vivo, if there are different isoforms that trimerize at the same, in the same trimer, and if if you have a missense mutation, how much might one missense with two wild type or two missense with one wild type, how much might that change function? Is that anything you can start to get at here, or do you mostly look at single proteins?
Absolutely. That's a really good question, and something we are actively thinking about in the exact same way. The original revelation that this could form a trimer was really fun and exciting. A lot of the work was done with a piece of that tail of Syngap, just for practical reasons. It's very hard to make the whole protein and to study those higher order interactions. It looks like that piece looks really stable if you purify that portion of Syngap. That research was really strong, and it looks like that trimer really can form, and especially in the purified setting, and it does seem to form that then higher order lattice work by interacting with PDZ domains of PST95. The in vivo part, though, is the real fascinating question. The postsynaptic density is full of things that compete for interactions with PST95 and the PTZ domains. And so it'll be interesting to ask questions of how dynamic this is. And I think it is clear that it is highly dynamic. That new preprint had some really fascinating stuff looking at competing phase transitions and sort of phases within phases. There's also like what happens when you phosphorylate in that region. Does that loosen or tighten up those trimerization interactions or the interactions with other scaffolds? And that is something that we're exploring as well. It's pretty straightforward for us to do that in a really reductionist setting, to explore like trimers interacting with other scaffolds and looking at the consequences of phosphorylation. But I also think it'll be really important to study that in a cell-based system, which is a little outside of our wheelhouse, but I'm confident that there are other labs working on that too. And in that cell-based setting, you would bring in the kind of context of the competing interactions, like how does this kind of dynamic competition of these same limited binding sites work, instead of just looking at it with 1 or 2 purified proteins, or even 3 or 4 purified proteins. The global PST is a much more complicated place. Oh, and I think you were asking about mutations as well. That is also something we are thinking about, and I'm intrigued by. From our broader experience with other signaling proteins, that trimer paper really did seem like it was quite a stable interaction, and it probably isn't quite that locked in the cell, and there's probably again aspects of having competing interactions that are involved in that. And I think if that were a little held together more loosely, which would be more typical of other signaling proteins, then you could easily see that being very vulnerable to perturbations like missense mutations. And I think that's also reflected in the sequence conservation of this through other organisms. It is highly unusual to have this degree of conservation in regions that could otherwise just be there for the flexibility. It does seem that that conservation hints at specific interactions with other proteins that would then have real consequences if they were mutated. Yeah. Any other thoughts?
Let me check Facebook again one more time real quick.
Sure. Well, there's a lull. I again wanted to thank you for the opportunity to interact with this community. I'm really, really in awe of what you are doing, and it's very, very exciting to get a chance to share at least the beginnings of our work in this area.
Thank you so much as well. Yeah, this is great. I don't see any on here. JR, do you have anything else? Or
No, I was chattering away on mute, but I
sorry.
during that poll, but
Okay.
but no, I think that's great. I mean, I think this is I think the main thing is we have a grant cycle on, for September 1st is when we accept grants for our on a 6-month cycle, September 1st and I guess February 1st. So if you want to write a grant to have a postdoc help you with this, please do. This is exactly the kind of work that we've been hoping for. We just didn't know how to say it because we didn't know any of this. But yeah, understanding the structure of Syngap and understanding how single nucleotide changes could all over the protein, how they could potentially be affecting things but I'm so glad to hear it. That I've heard that the domain of unknown function, but you're kind of calling it this unstructured, domain of unstructured is really, really interesting. So I'm really excited to continue to be in touch with you. I'm really glad that you told us about this at the beginning, and we're all rooting for you and your team to get lots of clean data.
Thank you so much. I was so happy that you all reached out, and it's great to make these connections.
Thank you.
Okay, well, everybody have a good day. Thank you so much.
Bye.
Bye.
Bye.